Hello, and welcome back to another episode with It's Dr. Dan. And today we're going to be learning about the structure of proteins. Specifically, we're going to be diving into all the different types of ways that we can classify and understand the structure of a protein. So, we're going to go step by step, building up our proteins, starting from the amino acid sequence, then going through all the individual intermolecular forces for how the structure can be organized to help stabilize itself, and then eventually we'll go into all the kinds of different interactions that can try to stabilize with the environment that it's around. So we'll go through it step by step. This is a very introductory biochemistry video, so it's not going to go into too crazy graduate level, senior level detail, but just enough so we can understand the basics. So let's take a look and let's dive into all these different types of structures. Okay, so the first thing that we're going to be looking at is the primary structure. So, what that is when we are looking at the individual sequence of amino acids that are bonded together. Now, when we look at one of these sequences, usually what you're going to be looking at is different structures that they could be drawn either like an expanded structural formula where it's very linear, kind of 90 degrees, or you might see it where it's in that zigzag skeletal notation. Personally, I like to draw it this way because it makes it a little bit easier to follow. So when we're looking at the backbone of it, the big thing is that it's a linear chain, meaning that it's in one continuous line. Now, when we look at that, you'll see that it's in the same repeating pattern. It always starts at the N terminal, right? So where the amino group is. Then from there, it's always going to go from left to right to where the C terminal is, or where the carboxylic acid is at the very end of that chain. Now, it always goes from left to right because each carbon that is in the middle of each amino acid is considered to be the alpha carbon, meaning that it has chirality to it because you have four different groups around each and one of those carbons. So each one of these where you see an R attached to it, that's where the alpha carbon is, like right above it. So we have a couple of them on this particular one. So now when we look at this, each amino acid is called a residue, meaning that each one is unique. It has potentially these different unique characteristics. So you see these R groups attached to them, meaning that they have these different side chains that are attached to each one. Now, when we're looking at the sequence, you'll see that it has this repeating pattern. So you'll keep seeing the same pattern of NCC repeating in the structure. So when I see that N, then you got C, C, so we got N, C, C, N, C, C, and it'll keep it going as it's bound together. Now, each of those different residues or amino acids, they're each bound together via a peptide bond. So you'll see I have these peptide bonds in green here. So just to kind of help highlight them a little bit. It's the same thing as an amide, right? So if you think back to organic chemistry, so that amide was the double bond O with the nitrogen, you're looking for the exact same thing. So if you're an organic chemist and you're more in favor of that instead of biochem, look for the amides. So being that we have that repeating pattern, the thing about that is that's what we consider to be the backbone of it. So that repeating nitrogen, carbon, carbon, that is where we talk when we say backbone, we're referring to that. Now, when we talk about those amino acids, the way that you can organize them is we like to describe them based on polarity, right? So if it's polar or non-polar, or if we want to switch to biochem talk, hydrophilic or hydrophobic. So are we loving water or are we afraid of water, right? How exactly do we want to look at that? After that, how acidic or basic is it? So is it something that has a carboxylic acid on it, or is it something that has an amino group on the chains? So when we're looking at those different amino acids, we can organize all the different amino acids by the different properties of it. So it all starts with hydrophobic, hydrophilic. So when we're looking at all of them, right, we have 20 amino acids that exist, right? So 20 essential amino acids that are essential for your metabolism. 9 of them are hydrophobic. So that includes glycine, alanine, valine, leucine, isoleucine, phenylalanine, methionine, proline, and tryptophan. Now, the hydrophilic ones, which are water loving, right? So because we have that love for water, right? They're polar. They can be separated based on their pH. So if it's acidic, that means that it must have a carboxylic acid attached to it. So when we have the carboxylic acid group that's attached to that structure, and we're talking about the R group itself, so where the special feature is, then it's going to be acidic because it has the ability to donate a proton. If it's basic, that must mean that when we think of basic organic groups, this has to have an amine attached to it. Okay, so basic has an amine, acidic has a carboxylic acid. So you're looking for those. Now, when it's considered to be neutral, that's generally anything that's just polar. So usually anything with alcohols, amides are the most common ones that you're going to be seeing. So if you're looking for any of those different things, like usually can do hydrogen bonding or dipole-dipole interactions, if you want to think of intermolecular forces. So talking about intermolecular forces, let's now dive into what kind of forces are these different amino acids capable and how can our structure continued to evolve when we want to look beyond just the order that these individual things are bound in. How can that change? Especially as the structure continues to grow and get bigger and bigger. So now the most important feature is when the chain starts growing, being that it has a geometry to it. So there's tetrahedral and there's trigonal pyramidal and trigonal planar geometries that are being seen in the structure. What's going to happen is going to start folding and bending. Now, and especially because this molecule is getting very large, right? If you're talking 10, 20, 30, 100, hundreds of amino acids long, it's going to start kind of folding and bending, and it's just going to get very, very long, very long. Kind of think of it as if you take like sheets off your bed and you were to tie them together, right? And you had like 50 sheets and you tied them all together. Well, eventually, how are you going to be carrying? If you wanted to carry the whole pile, it's going to lead to this giant clump of sheets. Well, it's kind of sort of a similar idea, whether that's a good analogy or not. It's following the same thing. So what's going to happen is the secondary structure, so the next level of organization, is as this thing folds, it can start hydrogen bonding to itself. And the reason for that is because on the structure there are amino groups, and then there's carbonyl groups on it, meaning there's nitrogen groups and CO groups. So what's going to happen is the amino group is a hydrogen bond donor, meaning it has the hydrogen part that's participating in the hydrogen bond, and it's going to hydrogen bond to the carbonyl on the opposite side. So if you kind of remember how we would label things like that, right? We'd say delta negative plus. So we have that acceptor donor relationship. So now that when this occurs is because that polypeptide that we're making as you increase amino acids is going to start folding on itself. And the reason it wants to do this is for stability. It's to help protect the protein and also serve as help its function as well. Every protein has a specific shape, so it does a specific feature, it can bond to specific things, it does specific things. So stability and function. So the way that these can look in a couple different ways. So the first one is in a helical pattern. So with this one, if you can kind of imagine this as like a kind of like a ribbon that's sort of like spinning around, right? So this is spinning, spinning, and spinning and spinning. So the alpha helix, the way that this works is essentially you'll have all those hydrogen bonds that are happening, where about every fourth amino acid, you'll have a hydrogen bond that's occurring in this chain. So you're going to see all these little pink little hydrogen bonds within the structure that's going to be forming between all those amino groups and the carbonyls. Now, as this is twisting together, it helps kind of it puts all the R groups on the outside so that way they can interact and kind of even do further interactions around it. So it allows it to have this ability to either connect to different proteins, maybe, or maybe potentially connect on different sides of it. So that way there's different shapes and different abilities that can form. So that's the alpha helix. Now, the other option that could occur is the beta pleated sheet. So that the way that this one works is it's kind of like a zigzag pattern that keeps kind of happening. So it keeps zigzagging, then it wraps around. And what we're seeing here is we'll see those hydrogen bond interactions that are occurring across the sheet. So that in order to help bring those together, so it helps protect it from anything foreign that could eventually take it apart. So we have these two different shapes that help do it. Now, the thing to point out is that this is between the backbone of it, right? It's not our groups that are talking to each other. It's only the pieces of the backbone that are interacting versus our next structure. So in our next structure, the tertiary structure, this is when the side chains are going to start to interact with each other. And the big idea here is that there's going to be this spatial arrangement of the amino acids that are going to happen. So once you start kind of having those spiral patterns, and especially when the amino acid gets really, really big, then you're going to have side chains that can interact with each other across hundreds of amino acids away because it's folding so much. And those R groups are polar as well. So what they can do is they can do all kinds of non-covalent interactions, aka intermolecular forces, where they can interact with each other to help stabilize. Now, they can also make bonds as well. So there's five major interactions that the R groups can do. There's hydrophobic interactions, meaning nonpolar. So think either dispersion, London dispersion, van der Waals forces, whatever you learned them by. That's what we mean by hydrophobic. Hydrogen bonding, right? So if you remember, it has to be an oxygen, nitrogen, or a fluorine attached to a hydrogen. And then salt bridges. So we'll talk about what that means. A disulfide linkage and a hydrophilic interaction. So now when we like to think about these, so let's start with kind of the first one, which is hydrophobic, and also kind of feeding into hydrophilic as well. So when we're talking about proteins, almost all of them, you got to think where are they located? They're inside your body, right? So inside the cells in your body, what are you mainly made up of? You're 70% made of water, right? So meaning your proteins are in a aqueous environment, meaning made of water, right? So what happens to those nonpolar molecules when you put them in water? Well, if you ever dropped oil into water, well, what kind of happens? Well, even if you try to kind of spread it across the surface, it starts to kind of clump together or aggregate together. And the whole reason why that's happening is, well, like dissolves like, the oil wants to be near itself, where it's considered to be hydrophobic as well, meaning it's really scared of water. It's like, oh God, you're different. You don't have the same polarity. We're not able to mix as a result. So when that happens, what we're going to be seeing is that the protein's going to fold in a way to help keep the non-polar chains, right? So the hydrophobic amino acids away from water. So what we're going to see is that the amino acids that are hydrophobic will be on the inside of the protein. All the hydrophilic ones are going to be on the outside. So the way that we can actually look at that, so if I have a sequence of amino acids, so here is our primary structure. What you're going to be seeing is with this primary structure that eventually it's going to get really, really big. And when it gets a little too big, you're going to see that it's going to fold. And now when how that thing folds, so if I start with the N terminal again, you're gonna see it kind of fold in a way where all of the non-polar regions are gonna be towards the center. So the hydrophobic ones are our yellow, hydrophilic are our pink. So what's gonna happen? It's gonna fold so that way all the polar ones are on the outside. So that way they can interact with water all around it, right? Because it's going to have a water shell all around that protein surrounding it. So what you're seeing here is on the inside is all those hydrophobic interactions on the inside. Nonpolar dissolves nonpolar or interacts with nonpolar in this case. On the outside, you have all hydrophilic. So it's like a shell of hydrophilic. So the way that we like to see the protein is that when it's in an aqueous environment, we have a hydrophobic core and a hydrophilic exterior or hydrophilic shell that forms around it. Let's look at our next tertiary interaction. So let's kind of draw those. So the first one with hydrophobic, the way that I like to draw them personally, and that is it's a very common way of showing it, is when we look at amino acids, we like to show how the R groups are interacting. So the way that I like to personally draw them myself to make it easy to see is I like to draw where the backbones are being turned on their side, and then we draw the R groups for our different structures. So right here I have phenylalanine and valine, and what they are doing is they have a hydrophobic interaction. They're both all carbons, hydrogens, meaning very, very nonpolar. So we see that these are going to be what's interacting on the inside of our protein. Now, when it comes to hydrogen bonding, what are you looking for? So this one, it follows your organic roots, right? So the way to remember hydrogen bonding, if you struggle with that, is if you remember the phrase F no to hydrogen bonding, right? So F no to that, meaning fluorine, nitrogen, oxygen. If they are bound to a hydrogen, they can be a hydrogen bond donor. All right. So when we look at these molecules, what we are seeing is, right, so we have on the left here, we have an alcohol. It can be a donor. On the right hand side, we have an amide. That one can also be a donor as well. But the way that we have to draw it, it has to be a partial negative, partial positive, negative, and then positive, right? It has to follow that order. So in this case, the alcohol is hydrogen bonding towards the amide group or the nitrogen portion. So we can see that interaction and we can label them just like so. So when we have a salt bridge or an ionic interaction, this is where the classification really matters when you're looking at your amino acids. So this one, what makes a salt? You have to think back to like general chemistry, introductory chem. How do you make a salt? Well, it's whenever you had an acid and a base neutralize each other where you make a salt in water. So whenever we are looking at these, the way that they're going to be working is that you need one of the acidic amino acids, which is aspartic acid or glutamic acid, and it has to be interacting with a basic amino acid, which is histidine, lysine, or arginine. Now, the way that that works is it's all about the acid base reaction. So, acids donate protons. So, what's going to happen here is that the proton that is on the left hand side from our acid, it's going to donate it to the base. So when that happens, right? So if we were to lose that proton, so when it loses that proton, it's going to be donated to the amino group on the other side. So the way that we are actually going to see this is you actually have a negative and a positive charge on both sides. So we'll have a negative on our carboxylate group, and then we're going to have a positive on the basic group. So when that happens, we are forming what is a salt bridge or an ionic interaction that's occurring between the two species. So it has to be an acid and a base. So it's that donation of the proton. So we see that we have an extra or an extra proton on our amino group that is here. So when we have that one extra proton that is being shown, it's donating that. And that's the key there. So whenever we show that, you have to show the charges because that's the most important. This is one of the strongest interactions that exists because we have that ionic characteristic, right? That is a strong interaction. So when that occurs too, so originally this was aspartic acid on the right. What happens is the second that it loses its proton, that's when it becomes aspartate. So that ATE ending is when we refer to when it's an ion. Okay. So if you ever see that and you're like, where did that A come from? It doesn't match maybe my references from my textbook. ATE tells you that it's ionic. It's a carboxylate ion. So just to kind of spell that out. And then the other one becomes an ammonium ion. So when we are seeing that change, right? So we have an ammonium ion on the right. Let's take a look at our next one. So with the disulfide linkage, the way that this one works is that it must be between cysteines. So cysteines can be cross-linked to other cysteines, meaning that it's going to make a bond between different parts of the different protein. So when this occurs, this is usually an extracellular chain that's occurring within these intramolecular forces that's in the molecule. So whenever we're looking at this one, when I have two cysteines, it's actually going to go through an oxidation reaction. One of the most common oxidations really with thiol groups, which is an SH group. So being that cysteine has thiols, what it's going to do, it's going to remove the hydrogens. So when it does that, it's going to create a covalent bond. So both hydrogens have to be removed and it's going to make an actual bond. So this is something that can really help stabilize and lock in the shape of our molecule when that occurs. So when it comes to the different folding that's occurring in very, very large chains. So for example, one of the most common ways that you'll see this one is like you already look at the structure of insulin It has multiple disulfide linkages that help structure it and help make it a very stable structure to be able to last quite a long time and be utilized within the body. So our last one isn't one that actually happens between amino acids. It's actually the interaction that we talked about earlier, which was, if you remember, when you think of how the protein folds, when we have these polar side chains, they're going to be pointing out a lot of the times towards the exterior of the molecule. Now, if you have a polar side chain, what it can do is, well, just like any polar molecule, if it has the ability to hydrogen bond, it can do it, right? This is in a water environment, so you're going to see water solvating anywhere it can because it's kind of sticky. It's going to get stuck on all these molecules. So anywhere there might be a lone pair or there might be a hydrogen, water is going to take advantage of that and have that hydrophilic interaction. So we're going to have this residual water solvating and surrounding all these different proteins. So that way they're able to be within solution and to be able to travel around the body, right? Can it be transported through the bloodstream? Can it be brought across maybe the cellular membrane? How can it try to be brought throughout the different regions of a cell? This is one way that it can help be kind of transported and carried throughout the body through a stream of liquid, right? Now, the last type of example is our quaternary structures. And this video really doesn't focus a lot on this one, even though it's in the title, but this one is arguably one of the most important categories as well, and it's something that can have a whole video in its own. But the way that we classify this one is when you have multiple polypeptide chains. So earlier, when we talked about primary, secondary, tertiary, those are all together. That was talking about one chain. So if you take many of them and you start kind of folding them together and bonding them together either through disulfide linkages or through ion or salt bridges or hydrogen bonding, it can create a super big complex that could have a variety of function. So here is hemoglobin. So on this one, you can see all the multiple different tertiary structures that are here and secondary structures. So you'll see that we have all of the different alpha helixes. So see all the spirals that are within the structure. You'll see that there's actually even some examples of, if you look closely, you'll see some like areas where there could be pleated sheets in it. And the thing that's showing here is there's multiple colored regions. So you see that there's the two red sections on the top left and top right, or sorry, bottom left and top right, and then the two blue regions. These are four different subunits all interacting together. So that way it can help bond and interact with iron, which would be kind of in the middle here, and it will help it'll be able to transport oxygen throughout the body. Okay, so you can kind of imagine this whole molecule is here just to transport O2 around your body. So that way you can be able to function and breathe and be able to do all of your functions. So many amino acids working together to help transport throughout the body. Okay. So this has been my video on the structures of proteins. I hope that this video helped you kind of understand and wrap your head around all these different definitions, and if you have any questions, please list them in the comment section below. If you have any other videos that you would love to see, let me know. And thank you so much for watching. If this is your first time here, please hit that like and subscribe button as always. And I hope you all have a wonderful day. I'll see you all later. Bye now.